Good evening, and welcome to Cognitum, a show dedicated to the present and future of science and technology. I'm your host, Iosif Gerstein. Tonight we have a very special episode filmed on-site at the Biomarkers of Aging 2024 conference, held at the Joseph B. Martin Center at Harvard Medical School, an event that gathers the brightest minds across science, medicine, and technology, united by the mission of unlocking and understanding the mysteries of aging. For centuries, the idea of extending human life was confined to myth, but today, thanks to cutting-edge research, we're not just asking how we age, we're exploring how to measure, manage, and even reverse it. Biomarkers, those biological signals hidden in our cells and tissues, are the keys to this puzzle. They're revolutionizing how we diagnose diseases, track health, and predict the future of our bodies. Over the next hour or so, you'll hear from the pioneers driving this revolution. Biologists, biotech visionaries, and clinical leaders will share their insights on how biomarkers could redefine everything from personalized medicine to the progression of aging. What does it mean to slow or even halt biological aging? And how close are we to making it a reality? This isn't just a scientific journey, it's a deeply human one. Behind every breakthrough is a vision of a world where longer lives mean healthier lives, where age becomes just a number, and where we have the tools to thrive, not just survive. So join us as we explore the frontiers of aging science, hear from the leaders shaping this field, and ask the big questions about the future of longevity.
I'm Max Anfried. I'm a research fellow at the National University of Singapore, and my research is on aging, lipids, and complex systems.
So could you tell us about your research?
Sure. So what I've been working on is you've heard about like all those different kind of clocks here, especially at the conference, and you have obviously different kind of omics. You have like metabolomics and proteomics, and a lot of clocks have been built on that. I've been working on lipids. And lipids is like this class of molecules that's like very heterogeneous. They're often classified as like being not really soluble in water. So butter are lipids, like that's your fatty acids, your oils. But that's only a very small subclass of them. Like your hormones, like steroid hormones, testosterone, estrogen are lipids. Sphingomyelin, ceramides. There are like more than 350 classes of them, and a few of them we have just been able to research them in the last few years because the technology wasn't available to measure all of them. And what I've been working on using those data sets that have like those lipidomics and build predictions for aging on them. What we mainly did is we actually took data from pathogenic cohorts, people with pancreatic cancer, and thought if we and try to build a model that can actually predict mortality of those cancer patients. With the idea is that maybe there's like similar dynamics in mortality between cancer and aging, and that once we have a clock train of mortality on cancer people, that could also used to predict age in healthy people. That was the hypothesis, and what we actually showed, you can indeed use cohorts full of pathogenic people, and there's like still a strong enough mortality and aging signal in there that actually can predict the aging rate and biological age of people that are healthy or have other diseases, and it all still fits like with the certain ratios that make biological sense. And what that in a way enables for the biomarkers field is most people only work on those small cohorts of 5 to 20,000 people that are kind of healthy, right? But what we can now do, we can actually go to the cancer people and tell them, hey guys, you've been collecting data the last 50 years. You have like this massive cancer databases, and can we actually have a look at them, or can we use them and build aging clocks on them and see then how well they work and what is actually the real connection between cancer and aging.
And how do you see the field progressing over the next 5 to 10 years?
I guess there are like two different kinds of the field. There's like the lipidomic field and there's like the aging biomarker field, right? I think lipidomics has a really good time currently. And if you think about like 40 years, 50 years ago, a lot of people were working on lipids. That's how we find the cholesterols and whatnot, right? Then there was like this time where not many people focus them anymore because technology was not there to, in a way, visualize them or detect them properly. And now the last 10 years, technology in measuring lipids has become much better. And now I would say lipidomics is where like proteomics was like 15 years ago. So like not much more funding is going in there, much more people enjoy it. So we will have more good data sets with high quality lipidomics. Obviously, relating that to the aging biomarkers field, where the trend is currently like into multi-omics clocks, right? That you not only use epigenetics or proteomics, but you try to combine them. And the biomarker consortium is talking about that as well to add metabolomics and lipidomics to their panels and get like a more holistic view of the entire process. And I think lipids have to play a role in there because we know they're like important for inflammation. We know lipid membranes change with age, right? And if you look about it, that without lipids, there would be no multicellular life because without lipids, you wouldn't have like a lipid bilayer, you wouldn't have like a membrane, and like in a way, like the lipids in this case, like a country that has a strong border, they have like provide a strong border for the cell, and there are like no intruders then that can harm the cell from the outside. So I think have a role to play within aging, and being able to measure them better is probably useful.
Is there anything in particular that has you most excited about the field of biomarkers?
Um, I think generally what is good at people start thinking or realizing that we need much bigger data sets, because the problem is always when you work with data, it's very easy to create a good model that's like 90 or 95% of the time correct, right? But if you really talk about medicine, you need models that are 99.9999% correct. It's similar to self-driving cars, right? You can't build your self-driving car that is like 95% good, but nobody would let it on the road, right? So to have the full product, um, getting to the level where you can use it, you have to need much larger accuracy. And I think people start realizing that. And I just had a conversation with somebody, and I said somebody probably should raise global funds, 10 billion, and say we build like a global database of like say 2, 3, 4 million people that are fully profiled, because then you actually have like a data set that kind of captures all the biology of all the different humans, different ethnicities, different eating habits, that actually then probably is representative and actually can make it to the clinic. And I would think that's actually the largest obstacle, but it's also like the largest opportunity or the most exciting thing to do. If you really get this data set that is so large that you actually can cover all of the population and build reliable models.
And if people want to learn more about your research and your work on lipid clocks, where would they find you?
I guess Twitter or X is the best, and it's at Max Unfried, unfried like unfried chicken. And yeah, you can follow me there, and sometimes I post about it.
Thank you for your
All
time.
right.
It
Hey,
was great
I also
speaking.
thank you very much.
My name's Nathan. I'm the co-founder of the Longevity Biotech Fellowship. And basically, we're a nonprofit that's mission is to get as many talented, smart people to work on solving the problem of aging. And we run these cohort fellowship retreats. And they start off with an intensive workshop in different places, usually like in Tahoe, but we've done a couple in Europe as well. And basically, we bring in industry mentors. So these are either scientists or researchers, entrepreneurs who are working in the field of longevity, and they run these in-depth, intense workshops on different bottlenecks in the space and how people can apply their expertise and skills to work on the problems, the outstanding problems in longevity and aging. And we also connect other people in this community with each other, because we think that one of the most important things is if you're going to dedicate a large portion of your time and energy and life into solving like a really important problem, you want to make sure that you're surrounded with other people who could be potential collaborators who share your same ultimate goal. And for us, that means, yeah, solving this problem of aging.
And what do you see are the key bottlenecks in moving aging forward or moving longevity forward and stopping aging?
Yeah, so there's a bunch. We actually, as a community at the Longevity Biotech Fellowship, did this survey report where we surveyed about like 400 people working in the field and just asked them, yeah, what are the biggest bottlenecks in your words, in the space in longevity? And we found that there was three main bottlenecks, but One of them that stands out, and which is pretty relevant to today, is the lack of validated biomarkers. So we're today here at the Biomarkers of Aging conference here in Boston, and so the fact that doing clinical trials to see what actually works to modulate the aging process could potentially take a long time if you're actually just going to wait to see how long people live or et cetera. So one of the ways that we could potentially shortcut that is by developing validated biomarkers that could detect a signal of an intervention working to extend healthy lifespan earlier. So that's one of the things. But then, of course, like a lack of public data sets, also just a general lack of funding in this space is also particularly of interest because, if you think about it, we live in this completely bizarre situation where it's known that aging is the largest risk factor for essentially most, if not all, of the major diseases, chronic diseases that kill most people. So like the top 10 causes of death, about like 80% of deaths are related to aging, right? And despite the fact that aging is this big risk factor, we spend very little amounts of resources, both human, financial resources, capital, etc., to actually try and solve this problem. Like only 0.5% of the NIH budget is actually allocated to fundamental research on aging biology, which is crazy, right? Because this is the thing that's killing most people. So that's one of the three major bottlenecks that we identified. Yeah.
And so what's your vision for the next 5 to 10 years of the Longevity Biotech Fellowship?
Yeah, so it's a great question. So far we've done 5 fellowship cohorts over the past 2 years that we've been operational. And we've seen a lot of great results so far from our alumni. They have been connecting with each other through this community and through this program, getting up to speed with the context and knowledge that they need to try and tackle this problem. They've started companies, found their co-founders, found funding. Some people have joined other startups. And so we've seen a lot of positive outcomes so far. But what we really want to do is scale this further. So instead of just doing these workshop retreats, which are just like, they're amazing because you bring in all these people from all over the world. They're just heads down for 4 days. Everybody's just like intensely talking about aging and bouncing ideas off of each other and getting work done. But what if we could not just do this for 4 days, but what if this was actually like a continuous, permanent base like of operations, like an actual physical place that we could do these sort of cohort retreats, but also longer programs where people can be in contact in a physical community, learning and creating and tackling this problem together. So ideally, what we want to do is scale this up to potentially some sort of, a new school of sorts, not like a university, but like a new place where ideas and people can come together to create new things in a more long-term fashion. So that's kind of like the trajectory that we want to go on in the next like 5 to 10 years. Yeah.
At this point, given that we're at the Biomarkers of Aging conference, what do you think is the single most predictive biomarker that you would use in trying to evaluate your own health status or the health status of others in relation to aging?
Oh my gosh, I'm not an expert, so you probably shouldn't take my word for it. So if you had to ask me right now, in terms like of a functional biomarker, I think this is just my guess, and maybe for what I would do personally, even though I don't do this, would be to measure probably like VO2 max, right? That seems to be the one that's most correlated to lifespan. But other than that, like I've never done a methylation test or anything like that, DNA, methylation, biological age clock. I think if you talk to most people who research these, they're probably not quite ready for individual, like definitely not like individual clinical use, let's say. But people are developing like a whole range of different clocks here and different biomarkers, and I think given more time, that we'll see some results, and hopefully a set of validated biomarkers that could help in either the drug discovery process or in clinical trials and approval processes. Yeah.
Great. So wonderful speaking with you, and best of luck with Longevity Biotech Fellowship.
Awesome. Thanks.
Thank you.
So I'm Matt Schultz. I'm the CEO of Ocean Biotechnologies. And we make genetic medicines for longevity.
So tell us what Ocean does.
So it was kind of based on this idea that information is the essence of life, and there's things that you can make by changing your DNA or delivering information that would be very difficult to make through traditional means. So we started initially in senolytics, actually, so killing damaged cells based on their transcriptional activity. But our lead programs now make you build muscle for frailty and selectively ablate fat.
And how do they do that?
The muscle building one overexpresses a gene called follistatin, which is basically the body's biochemical pathway for skeletal muscle exercise. And the fat killing version uses an inducible caspase to selectively kill those cells based on their transcriptional activity.
And what is the product itself that overexpresses follistatin or caspase?
So we use a really novel or interesting delivery technology called a proteolipid vehicle. This is basically a lipid-based non-viral delivery technology that has a really unique fusion protein in it. It allows it to deliver DNA directly to the cytoplasm of cells. And so the treatment itself is basically a program written in DNA that we encapsulate in those particles and inject, and it will deliver it to the cells and express a protein.
And can you control the level of expression? Is there a dose dependency that you've found?
In the versions we've been using weren't particularly sensitive to dose of expression. Like the suicide gene is a very binary type of outcome. It either makes it and kills the cell or doesn't. And with follistatin, when you exercise rigorously with resistance training, the levels in your blood will go up about 7-fold. And so it's a pretty significant increase over a large organism. And we're mostly trying to push as much as you can. I don't think there's much of a downside to it. But it's relatively easy to modulate the dose on it. If you want to control how much any given cell is expressing, you need to build more complex logic into the construct itself to do it.
Mm-hmm. And what kind of results have you seen with these therapies?
Well, in mice, we just published a paper in Cell that showed if you gave them one administration of this, in 6 months, they're 50% larger and twice as strong. So it is really quite significant.
That's for the follistatin overexpression. And what about the fat ablation?
The fat one, when we started that work, was in human tissue explants. So basically, after someone had gotten a tummy tuck, they'd take the skin from it that they were going to throw away and punch it out and send it to us, and we could inject it. And in that case, with one treatment, we saw about a 20% reduction in adipocytes without any obvious damage to surrounding tissue with histology. And so that treatment in general is newer in development, and we've put it into mice and shown that we can selectively target those sites with histology, and it's now going into pigs actually. So that's we're sitting there waiting ourselves to see how it fares in that model.
To see if you can get fat pigs to be thin?
yeah. It's not going to be something the bacon industry is going to like.
Very interesting. And where do you see the technology of your company progressing over the next 5 to 10 years?
Well, when we put it together, like the field of use for all the technologies it's based on, we were really looking at two things. One is what could we build in the short term that the FDA would approve or that a pharma company might license. But the other was, where do we think the field's going to go in the next decade? Because as we're kind of watching this space evolve, there's a bunch of stuff right now that we don't really fully understand. The science isn't quite elucidated on it. And at some point, we're going to know these things better, but they're still going to need delivery. And so we wanted to build a platform that would allow us to take advantage of the field as it matures. So, we've been working with all sorts of interesting things, ranging from, the partial reprogramming to modulating the extracellular matrix. And I think the sky is kind of the limit on it, but we're trying to develop this to be in line with where the field's actually moving itself.
What has you most excited about the future?
Well, that's a tough one. I think what's really exciting, is that we are beginning to gain the ability to manipulate systems that we barely understand. And the field has progressed in such a way that most of the tools we're using today didn't even exist a decade ago, or at least were very nascent. And it's been really exciting to watch the world of genetic medicine and longevity really move from being a very obscure thing in science to something that is gaining lots of momentum. I mean, when this guy got started in the field, there were no approved gene or cell therapies, and now there's thousands in clinical development. And I think with longevity was largely kind of banished to the the sides for a long time, and now I think people are recognizing that these are systems that we have the tools to manipulate. And I think as we get better and better understanding of it, we're going to be able to live in good health for much longer. And I mean the idea that you're not going to be subject to constant decay of everything, or at least can push it off for a long time, I think is really exciting.
Well, given that we're at the Biomarkers of Aging conference, what is the single biomarker that you would right now put all the chips on in terms of having the most bang for your buck when it comes to evaluating longevity or how gracefully you're aging?
Oh gosh, I am not qualified to answer that one. I think when I got into the field, one of the biggest limitations I saw was that we had no good biomarkers. That I mean, no one's going to be running lifespan studies in humans. I mean, at the very least, your patent's going to expire before you see what happens. And so we just lacked the ability to see it all. I mean, even when we were starting in Senolytics, like couldn't even really measure SAS effectively in the blood. And the field's progressing on so many of these axes. It's really impressive. I mean, I think conceptually the idea of really being able to see if you can affect the trajectory of age or reverse it. The idea of a biological clock or biological age is, I think, really super valuable. But as you've seen from the speakers here, there is a huge range of things that people are playing with, and I don't know which one's going to come out on top, but I'm super happy to see them all doing it.
Well, it was great speaking with you. Thank
Awesome.
you.
Thank you.
I'm a PhD at Yale University and I study human aging. Specifically, I build biomarkers of aging to quantify how different organs in our body are aging from a single DNA methylation blood draw. And then we can really use these biomarkers to potentially build new medications that might slow down aging. And that's kind of what my research is really on.
And why is this important?
Well, the reason why this is important on a broader scale is that we know that we have an aging population. We would like that hopefully we can build medications to allow people to live longer, healthier lifespans. But on a more simplistic level, to be able to do this, we first need to be able to essentially build biomarkers that can measure how quickly a person is aging. But more importantly, if we actually have to run clinical trials, to run a clinical trial for aging, you cannot actually run a clinical trial for aging at this point because that would effectively take 30 years because the endpoint is mortality or death. So to counteract that, you need to have proxy biomarkers. Proxy biomarkers that can tell you that, whether an intervention is working or not. And that's what really I'm trying to do. Build these biomarkers that can really allow us to tell us whether an intervention is working or not. And that would allow us to essentially build these interventions and clinical trials that can run very, very quickly and in a very cost-effective manner. So instead of running a clinical trial for 30 years now, we can run a clinical trial maybe in 2 years at a much cheaper price. And that would hopefully get us an intervention much quicker.
And how far off do you believe we are from such a set of biomarkers that would enable fast clinical trials on aging specifically?
I think we are not too far off, in my opinion. So we already are seeing a lot of aging biomarkers from proteomics, DNA methylation, metabolomics that are performing really well in sort of what we call as 3 categories. One is prognosis, which is the ability to predict future diseases and future mortality. The second is responsiveness, the ability to respond to intervention. And the third is reliability. That means that they do not significantly change, say, between one test to another. And that's the exciting part. But there is scope for improvement. So we really need to potentially even improve these biomarkers further before they can move into trials. The second thing is we also need to validate which of these biomarkers go into trials. So here's the idea that before even we move them into a clinical trial, we need to run what we call as a surrogate endpoint trial. Now, this might sound a little complicated, but what we really need to do is run a clinical trial for a biomarker. We need to see that whether one of these biomarkers is actually predictive of change, of mortality, of diseases, and response in a clinical trial setting. So, for example, we take a biomarker for aging, right? Give it in a very well known intervention like exercise, which we know will hopefully slow down aging, right? Or calorie restriction, which we know will hopefully slow down aging, right? And then say, does it actually track with other outcomes of mortality and disease, disease occurrences and frailty, so on and so forth? And if it does, then we have a surrogate endpoint or a surrogate biomarker, which is great, perfect. And now we can use this biomarker in actually clinical trials and give it to patients all across the globe to track their health, and which would hopefully transform preventative medicine. Because now you essentially have an aging biomarker that works for all practical purposes.
What has you most excited about the field over the next 5 to 10 years?
I think what's most exciting to me over the next 5 to 10 years is the ability to do a lot of these analysis. We are generating so much data through these longitudinal cohort studies, through these small interventional studies, that we can really now look and do systematic analysis around these biomarkers, around these interventions, and how they are really changing human lifespan and health span. So we can really start drilling down on what's working and what's not working. So I do believe we are still like maybe 10 years away from getting our first gerotherapeutic, but at the same time, we are really moving forward towards at, I would say, light speed. Because at this point, we really have tons of data to really start validating all these biomarkers and interventions that we have had on the back burners for such a long time. And that's the really exciting stuff, in my opinion.
Well, given that we're at the Biomarkers of Aging conference, if you had to put all your chips on a single biomarker right now that you think has the best performance in terms of measuring aging or predicting the speed of aging, whether people are aging gracefully or not, what would it be?
I don't think I can put a bet on like a single biomarker. I think that would be a disservice in a way. But I would say that there are definitely sort of platforms of omics that I feel are more powerful than others at this point, looking at the data. So something like DNA methylation right now, we have been able to systematically see that it's prognostic, it seems to be responsive and reliable. We haven't done the same analysis for proteomics, we haven't done the same systematic analysis for metabolomics and for clinical biomarkers, at least in the context of aging. Until we do those systematic analysis, we won't be able to at least say that those platforms or those omic data types are sort of ready for clinical surrogate endpoints. So what I'm saying is that I don't think there's going to be a biomarker, right? I'm guessing there's probably going to be multiple biomarkers. But what we really need to do is validate the platforms first, right? Whether the platforms are ready, and then validate the individual biomarkers. So my bets at this point seem to be on DNA methylation, partly because I work in DNA methylation and that's where I'm biased as well. But that's also because it has probably had the most progress at this point in context of aging biomarkers. And that's I guess, the situation we are in.
It's very exciting.
Yeah.
It's great speaking with you.
Thanks for the conversation.
Thank you for joining us today. We hope you join us next time as we examine and elucidate the frontiers of science and technology with the thinkers creating our future.